it's Michelle Morand, cancer patient, educator, and advocate, specific to leading edge technologies, diagnostics, and treatment, otherwise known as precision cancer medicine. I'm here today to introduce you to Ross Cagan, Regius Chair of Precision Cancer Medicine, professor at Glasgow University, and a specialist in the lab specific to certain genetic technologies. He has partnered with a company called Vivan Therapeutics that is doing some really innovative work to help identify treatment options for cancer patients. I'm going to let him tell you all about it, but it's really interesting stuff what they're doing, and what it can lead to for every cancer patient, I think, is really exciting. Ross is so incredibly knowledgeable about precision cancer medicine, and specifically in the lab, and the type of work that these folks are doing at Vivan really is leading edge. So I know you're going to really enjoy this discussion because he's such an interesting, engaging fellow, and what he's talking about is so relevant if you are a cancer patient, an advocate, or a healthcare provider in any way. Knowing about these technologies, what's accessible now, and what's coming very soon for all types of cancers, I think is really important. So please enjoy this interview with Ross Cagan, and please put your comments in the chat. I would welcome hearing your thoughts on this interview and any questions that you have for Ross. Well, hello, everybody. I'm Michelle Morand, precision cancer medicine educator and advocate. And today I am here with Dr. Ross Cagan. He is a colleague at the Vivan team. Vivan is a very unique company that is providing cancer patients, specifically at this time, I think colon cancer specifically is the focus. Which is a very difficult cancer to treat, but they're providing a very unique service to colon cancer patients. So we're going to hear a little bit about Dr. Cagan's background, and specifically, we're going to focus on what he and his team at Vivan are doing, and what he sees coming down the pike for precision cancer medicine and for cancer patients. Dr. Cagan, nice to have you with me. Thanks for being here.
Hey, thanks for having me on.
Thank you.
Should be interesting.
It will be, absolutely. So, and people will really benefit from knowing a bit about you and about what your team is doing. I have no doubt about that. So let's talk a little bit about you, though. Tell me a little bit about your background, and maybe a little bit of the story about how you found yourself doing this kind of work.
Sure. Always happy to talk about myself, I guess. Like all scientists, what we really like to talk about is our work. I come from actually a different background, developmental biology and genetics and so on. And long story short, we decided that the tools that we and others in my world have been building would work for cancer. So we took on a small cancer project that ended up resulting in a drug that was approved for a rare type of thyroid cancer. We worked with a company, AstraZeneca, and I thought, wow, that went better than I expected. So moved the lab from St. Louis to New York, and we began to think more deeply about, what are the problems in cancer? Since we're coming in from left field, maybe we can rethink some of the assumptions. So the first thing we asked is, we keep curing cancer in our labs, but it doesn't always turn out so well in actual patients. Where's the disconnect? Mm-hmm. So that was the beginning of a journey, which I will spare you the details of. But it led to us thinking about using fruit flies, Drosophila, to capture the complexity of the disease. This is the key. So when you work on more complicated animals, you can only change one or two, or maybe if you're really ripping, 3 genes. In flies, we change 10, 15 or more genes. And what we found is when we started to do that, drugs that worked in the simple model stopped working. And we thought that's a good sign because they weren't working in patients. So we thought we would be onto something. And I'm going to skip a whole lot of stuff and cut to the chase, which is at some point we realized we've done a lot of work, my lab with others. We did a lot of work to show that this has value, but at the end of the day, It really comes down to whether it works in patients.
Yeah.
And so we began a clinical trial back in New York. And that clinical trial, in that clinical trial, we made for each patient their own personalized fly avatar, where we changed many genes. And then we treated the patients, including colorectal cancer. We also took on some other cancers. And actually, that went very well. Not perfectly, we didn't cure every patient, and it would be, it's important to note that it's experimental. But the main problem was we started out well, we started out fast, we had some responses in cancers that didn't respond to really anything. And then the pandemic hit and shut the trial down. However, and then I moved to Glasgow, and where I'm Regius Professor of Precision Medicine here, and also working with Vivan. I'm grateful to Vivan because they had picked up that baton and have run with it. And unlike us working out all the sort of initial ideas and so on, what Vivan really has done is brought a higher level of technology to it. So they have really leveled it up, the approach, and they're building a database from all of this work so that someday, I mean today, but someday all patients, you won't need to go and have every patient have their own avatar made. Right now, that would be the best to make your own personal avatar all the way out. And it's pretty unique.
Yeah.
But of course, what we really want to do ultimately is crack the code, but that hasn't happened yet. We and others have tried.
Yeah.
So that's work in progress.
Obviously you need bigger numbers, you need more cases and
Correct.
a bit
That's
more
right.
time. I totally get that. We see that with our liquid RNA too. That you do start to see themes though,
Yes.
absolutely, that
Yeah.
and that's what leads you to say, I think we can create kind of an AI or a database that allows us with some a patient sample or some data about what's driving the bus for the patient. We could then match them to what we already know. Yeah, but we're getting ahead of ourselves, because for those of you at home,
Uh-huh.
let's back up the bus and talk a little bit more about what Vivan actually does. Tell us a little bit more about these fly arm, like why flies? For starters, you said, in comparison to mice, which you can change maybe 2, maybe 3 genes with the flies, you can change 10 to 15. Why is that? Tell us what about fruit flies that makes that work?
Sure. So I come from a long lineage of fly geneticists, fly biologists. This goes back for well over a century. Um, there have been 6 Nobel Prizes given to fruit flies. So it is, but it had not been used as much for disease. It was mostly to understand the fundamentals of biology.
Okay.
And that's what it's famous for, at least in my world. And so we're really bringing a century of those tools to here. But 2 things to know about to your question. One, you can manipulate flies quickly and easily in a way that would be very difficult to manipulate, let's say, a mouse or a monkey. And part of that is because fruit flies from egg to adult is about a little over a week. And for mice, it is far longer. And so if you make a change in the DNA, that's cool, but then you have to make a bunch of mice.
Right.
So when a patient is screened with their own fly avatar, they'll have typically about 400,000 flies that are grown up, that are their personal fly. So you build a fly and then you go to 400,000. So if you do the math in a mouse, it will take you centuries or certainly a century to grow, I think. In flies, we can do that in weeks.
Yeah, and
So that is that time frame. So between the ability to manipulate them and grow up so many of them, and then we break out the robots and we screen every possible drug combination to go after it. And this, because we're now doing this in flies, it allows us to think bigger for the patient and really leave no stone unturned. We don't make assumptions about what will work. We simply test everything. And that's the magic of that approach. The way I look at it is the flies are like a soft computer. You put the patient's data in, and it gives you the data back.
Right. And so where are you getting the initial data? Has the patient had some sort of tumor testing or what's happening? Yeah.
Yeah, so you're asking all the right questions.
Okay.
So a patient would come in, okay, and if Vivan felt they were a good candidate to help them, so in other words, the avatar approach would make sense for them. They would, either the patient very often already has their tumor analyzed for its DNA content, or that would be done.
Okay.
And then once that is done, that information, the scanning of the DNA, that information, so what causes cancer is changes in your DNA.
Mhm.
And so you would say, okay, here's 10, 15, 20 genes that are changed in that tumor. So normally you would just look at those, pick out one of them, one of the changes, and say, I'm going to come up with I'm going to give you a drug that hits that. So that doesn't work so well. And this is why I've gotten into this. That just has not worked well.
Yeah.
So instead, we'll just put everything into the fly. You have 15 changes, cool. They all go in based on that analysis of your tumor. And there are other things to consider. When you do the screening, we find drug hits. Maybe the patient would not, has a heart issue, so we would stay away from drugs that would affect the heart. So there's a lot of things that go into making a proper decision for each patient. And we have special tumor boards that help make sure that we're making those correct decisions. And only other thing I'll add to it is the other beauty of this approach is when we do the screening, we don't get a single hit, a single cocktail of drugs. We get a set of ranked drugs. So if you don't respond or you respond for a while for the first cocktail and become resistant, which is really common in cancer, cool, not cool, but we do have the second round, the third round, yeah, and so that discussion, so you can immediately move to those.
Yeah. So let me just make sure I get this. So
It's a lot.
there's all I appreciate that you can't necessarily help everybody. Um and if somebody comes to you, they've either already got some DNA sequenced or you would obviously help coordinate that. You take that information and then you take the fruit flies and you modify the genetics of the fruit fly to give them essentially the same mutations that the patient is showing up with. And then you try a variety of different drugs on the flies.
Mhm.
And within a period of how long from that point until you have a sense of, okay, here's our ranked suggestions. Here's what your flies did really well with, and here's what didn't help them.
Sure. Well, first, let me just note that there are different levels. Okay, so sometimes we'll make the patients fly, and we'll immediately go to certain hits because we already have experience and say, well, this set of mutations tend to respond to this. So we'll do a quick check with that. So that can shorten things quite a bit. In the fullest case scenario, if the patient has the time and if we think that makes sense, essentially every FDA approved drug, all of them are screened. So we don't think deeply about it in the most extreme case.
Right.
And then to answer your question in terms of timing, that is affected. The best way to think about it is weeks to months, depending on how far out first of all, how well the patient's doing, of course, and also, so how much time the patient has reasonably, and also whether we already have experience with similar ones that can shorten that. So it can be from weeks to months, depending on all of those.
And that is the point, isn't it? With precision cancer medicine, every case truly is unique. It really is. So you might have some data, as you're saying, you're getting to a point, if I heard you correctly, where because you've run so many experiments / you've had so many cases where you've built up these armies, you have kind of a short list, maybe when you see certain genetic predispositions or molecular features, you have a kind of a short list. Let's definitely try this. Let's definitely try this. And that can really help expedite it. And that I wholeheartedly believe is only going to increase the ability to do this matching. But I also love what you're saying too, because cancer is unique to every person, casting a wide net is critical. we don't want to make assumptions that because this works, nothing else will, or that's all we need to look at, or and I love that your team is doing both. You're synthesizing the data you already have to expedite some immediate options, and then you're also still running the full experiment, essentially casting the full net to see what else. So ideally, that first option works really well, but then you've got very confident backups, next steps.
Yeah, so first of all, Michelle, I could not have put that better. That's actually really well done. I'm more than impressed. Second of all, just to build on your points, we're entering a new era of cancer. So I'm the Regius Professor of Precision Medicine here at University of Glasgow. So I think a lot about precision medicine, and I'm sure you've heard all about precision medicine. It's almost like a buzz phrase at this point. But there's a problem with precision medicine, which is right now we're much better at gathering information than knowing what to do with that information.
Yeah.
So as you said, we can sequence the genome. The cost of that is tumbling
Mhm.
rapidly. It's now in the hundreds of dollars, which is crazy considering the billion plus that originally cost. So that's tumbling down. So we can, and there are other things what are called spatial omics and so on. I'm involved in a lot of this stuff. But that's lovely, but that's just a wall of information.
Yeah.
And the question is what to do with it. So some people say, well, I'm going to, computationally, I'm going to model this. The problem with computational is computational can only do what we already know is important. So you tell the computer, go find patterns that look like this. But since in many cases, we simply have no idea, like in colorectal cancer, which has been really frustrating, that's not helpful. So what do you do with all that information? And I think the brilliance of this fly avatar approach, if I may say, because of course it's close to my heart, but the brilliance of it is it matches that blizzard of information very nicely. Instead of throwing out most of it and using our biases, very much what you just said, instead of just using our biases and cherry picking what we think is going to be important, we finally have the ability to take that wall of information and use it to rebuild the patient and then just let that take us to an answer. I don't have any emotion behind what the hits are. And we have treated patients with some really surprising combinations that have worked. And then only then do we go back and try to figure out, okay, why is this arthritis drug helping this patient, right?
Yeah.
So it's that.
Yeah. So it's about kind of getting out of our own way. If we think about, I really feel for the oncologists on the front lines here when we're talking
Yeah.
about cancer, because they're really are stuck between all of these types of exciting developments that are kind of new and improved and have all sorts of good efficacy and mechanisms and all the things that really show that they are legit, and what their cancer agency tells them they can recommend and do. And there's such a chasm between these two things.
No, absolutely. And one of the lovely things about so the beauty of of using these little fruit flies is they're not wildly expensive. And of course, to grow up 400,000 of them, if they were expensive, would be a problem. And I think access, of course, is a big issue. So basically, this approach is we've really thought about this quite a bit. So for example, one of the big problems with cancer drugs that I only really learned to think about when I started to talk to patients who have been amazing in explaining to me what it's like to be a cancer patient is, these drugs are nasty. And it's not a great win if your quality of life is really poor. And I got a dose of real detail where cancer patients have taken me out to lunch and really explained me what it's like to go through this. And one of the big reasons we developed this was not just to come up with another way to batter cancer, which of course is important, but to do it in a whole animal where if the animal doesn't thrive, then the drug combination doesn't come up as positive. So we're looking for something where the animal thrives while the tumor shrinks.
Yes.
And I've really come to appreciate that this is an important part, this bit about quality of life.
Yeah, that we're experimenting on the flies and not the person.
Yeah, that. I do feel bad for the flies, but yeah,
Well,
that.
yeah, they still get a week of life or so, they're,
They
generally.
are pretty short-lived anyway, so it is all true.
Yeah, I know there is, flies, mice, whatever it is. There is truth in, the human world that we really are interdependent with all the creatures. And in this
Yeah,
case, 400,000 flies must give their life, but we are potentially saving a human life in the process.
that's right. That's
Yeah,
right. And hopefully we're saving the flies as well if when things go
yes,
well.
a good chunk of
But
them.
yes, all of this. And so I think that I and Vivan, I can, if I can speak for Vivan in that, we have really grown in that sense that we really tried to listen to the patients and understand what their needs are. And all of this is really designed with this in mind.
Yeah. And I know you and I had a conversation maybe 3, 4 years ago. I don't remember when, in anticipation of this conversation. And then we had the pandemic, as you say, and all sorts of things occurred. Um,
Uh,
Set aside. And I know that in these interceding years, um, your process has really been refined, and your like everything that you're talking about here in terms of how you now have this larger database of information that helps you at least start to get in the ballpark of certain combinations that might benefit somebody, so you can more rapidly test their avatars for those particular combinations. Yeah, and that's only going to increase this ability to kind of start rather than this vast universe of possibilities. Start at least in the ballpark so that we can get to the bullseye a little bit faster for people.
of course, time moves forward and we just get better at everything. So I won't bore you with the details, but a lot of the technology has simply been improved because when we were running the original clinical trial, we were like building the plane while we were flying the plane. So there's been a lot of technological advances. We're better at making these flies, the royal we. They're better at scoring the flies. And also during that time, um, sequencing technology has improved. Everything has gotten better, basically, bottom line. So first things first, just the time it takes to go through the process has shrunken. And that itself is an important thing for the patient, which means that we can think bigger for more patients instead of only some being able to go take the process all the way out. The second point that you bring up is absolutely the case, which is, and I have to say, I really have to give a shout out to Laura Tower, who's really been brilliant in this, that they really have leaned into this technology to begin to, as you say, create sort of hierarchies of drugs and drug combinations that you hit first. If none of them work out and the patient has the time, okay, cool, now we open up and go through everything. But of course, it's important that you be able to get there quickly because the sooner you can get back to the patient, the better. And
Yeah.
to answer your question is absolutely, as time goes on, that database gets refined.
Yeah.
And so that's another piece that just is getting better over time.
Yeah, that's very exciting. And so, also, I think you were inherently saying this in what you've seen. When you started this work, we didn't know there were over 550 cancer related genes, right? We knew if, I don't know, when you began, maybe 100, whatever, maybe a few
Right.
hundred. Now we know, at least kind of the better DNA tests, tumor DNA tests out there look at least 550 known cancer related genes. So that's how many there
Mhm.
are at minimum, and of course more being identified as we go. And not all of those have targeted therapies at this point, but many do. Are you keeping up with this very new, exciting side to things of RNA? Is your team exploring that at all?
Yes, so this would be, Michelle, we're doing so many things.
Yeah, I bet you are.
It's
Yeah,
wild.
yeah,
And of course, this information flows through to Vivan. So to answer your question, um, so as you say, our knowledge of cancer genes has come a long way since I first cracked that open. And we've even explored the non-cancer genes, putting those in and seeing they sometimes have impact, and we're learning quite a bit about that. We don't worry about whether we have drugs aimed at them, because when you have one mutation, you can hit it with a drug. But when you have 15, who knows what drug will, but usually that drug for that one target won't matter because things are complicated. To answer your question, however, not only have we walked deep into RNA, what's called transcriptomics. There's going to be a whole bunch of words like that, so I apologize.
No worries.
So RNA, protein, proteomics,
Okay.
and there's a new game in town called spatial transcriptomics, spatial proteomics, which we just gather and call it spatial omics, which is not just I pull your tumor out, I grind it up, and I find all the changes in it, but actually I take a section that has your tumor, has the surrounding tissue, has your immune system, and so on, and I go probe for all the RNA that's made in your body, almost all many of the proteins, and not just what changes, but does it change in the tumor? Does it change in all of the tumor? Does it change in the neighboring normal cells? So now we can do all this with high resolution, and we're doing all of this. And I think if I can, since this is the topic here, what I've really come to appreciate again is the brilliance of working in a whole animal for screening, because what we're learning here is that you have complicated interactions with your body and the tumor. And nobody's surprised to hear that. But actually what we're finding, to your question, is that affects drug response. And I think that may be the single biggest reason why it has struggled in clinical trials, is that whole body complicated interactions. Almost all drug discovery has been, I take cells, I put them in a dish, I throw some drugs on them, and if the cells drop dead, cool, now I'm going to go into clinics.
Right.
Not so great. That hasn't turned out great.
No.
So the ability to capture RNA, protein, but thinking about it in the whole body context is really, that's, the next generation of it, how we're going to be thinking about cancer. That's basically just hitting the scene right now. And yeah, I've come to appreciate how these fly avatars have really captured that in a way that I don't think we had thought deeply about when we were first making them. But let's just say this is a extra big bonus.
I'll say, yeah, I can appreciate that, because the real live expression,
Yeah.
is of the gene is, which we get RNA expression information from the tumor sample, absolutely, and that definitely helps us get closer to that bullseye. But seeing in a live critter, is there a desensitizing mutation? Are there things getting in the way of the treatment that should be working, and what the heck's going on with that? And being able to
Yeah.
assess that, that again, it's just going to help you get closer to the bullseye for every patient faster, and that's what it's all about, really.
Yep. Absolutely.
Yeah.
Absolutely.
Or is your team, when you're growing up these flies, are you using some of the new drugs like antibody drug conjugates and things like that as well? Or are you still, with all due respect to how advanced it is, are you working at the standard targeted therapies, or what kinds of things are you throwing at these flies?
Well, so again, so I think we're mostly aiming at drugs. Those are the ones that are most straightforward to make combinations.
Mhm.
I can tell you when you have a 15-hit fly avatar model, that it's rare, if ever, the case where a single drug is going to stop that runaway train, whether that's an antibody drug conjugate or anything else.
Yeah.
And so the only proper way that we can get the NHS or the FDA or any of these bodies to buy into combinations is to not take combinations of exotic drugs, but take combinations of drugs that have a long history to them, so that because almost every drug combination has been taken by somebody at some point. Okay, so we know reports, oh, this drug should not be together with that, those. So it's really important that you do this sensibly. We do not want to start hitting the patient with all sorts of crazy combinations. As cool as that sounds, you actually want to be, smart about it. So no, we stick with the FDA-approved drugs, but the key here is because we're creating these, new combinations, it's like a new drug, if you will. But
Absolutely.
it sort of addresses the whole body problem, not just see target, hit target.
Yeah, that's right. So the fly avatars help you identify the combination, and I'm right there with you. It's just never one mutation. It's never one drug, really. Although, things like immune therapy can provide significant benefit and a curative response
Mhm.
for some folks, absolutely. So we'll put that over there in its own little basket for those who are candidate for that. But otherwise, yeah, it's going to be a combination of three or so therapies. And then that takes care of those cells that are driving the bus. And then maybe in a little while, we need to add the next level, the next combo of 2 or 3 therapies. These targeted therapies, though, and including the antibody drug conjugates, many of which are FDA approved, they are, in my understanding, and I'm curious for your sense of this, they are better tolerated as a rule than standard chemo. What is your understanding of such things? Sure.
Antibody drug conjugates are a bit of a mixed bag in that, so for those who aren't familiar, you're basically using a tag that guides the drug to the target.
Yeah, that's
The
right.
problem is the tag goes and looks for a protein that's common on the cancer cells. But of course, not all cancer cells are going to have that because they're cancer cells. Some hide and so on and so forth. So it's usually a bit of a mixed bag. There's also versions of this where you actually carry a poison to the cell.
That's right.
Those are definitely not less toxic. And so you have to sort of calibrate them. Antibody drug conjugates are lovely and they definitely have their own world. And we can certainly use those in combination
Mhm.
with what we're doing with other drugs.
Which is more targeted.
However, I find it is going to be challenging to, for example, convince the FDA to buy into using antibody drug conjugates plus other drugs. They already consider that to be a combination. And if you look, you'll see that there's almost no examples where that's used in a cocktail. So I'm sort of simplifying things, but to get to that point. So again, we try to lean in on things that have, so the sort of regular FDA approved drugs, which increases very slowly,
Got it.
has a history to it. And, Laura can weigh in on this. Personally, I'm more comfortable with creating novel combinations with things that have probably been in people already. Because the thing I really do not want to do is take somebody who has cancer and batter them with some weird combination that's not well tolerated. So that is just sort of my preference. And maybe in the future, we will consider moving forward with things like drug antibody conjugates. There's all sorts of cool, new, exotic approaches.
Yeah.
DNA, there's the CRISPR world, which is just starting to hit and will be pulled for cancer. There's the RNA world that I think those companies that were so amazing in COVID, as you probably know, are now coming in with those. And all of these can be tested in flies. But I think right now, today, having that, I don't want to push that out to people and say, yeah, we're going to start throwing all these cool exotic things at you in the hope that they don't act together in some way that will harm you.
Right. So I appreciate that because that's interesting that the thought is the FDA will see an antibody drug conjugate as already a combination therapy.
Yeah.
That's an interesting thought. And fundamentally, what you've been saying all along today is we are here for the individual patient. And what we're interested in are options that are going to work well for them that their doctor will prescribe. That's
Right.
the key. And so we can't be experimenting and we can't be guessing and we can't be hoping. And that's where the Avatar Army is so helpful because you can see how many are still kicking and how many are vibrant and doing their thing and how many are it's okay but not great, and that I'm sure there's more precise measures you use to assess the benefits.
There is, but I get your point.
Yeah. So
Can I just say, Gina, I apologize. I just wanted to jump in on that very point because For me, and I think probably for Laura, this has all been a journey. So of course, as a scientist, I'm like, let's take the craziest,
Yeah.
combinations and just try, okay. But I have grown, like I said, by talking to patients. Sometimes when you start out with cancer, some of the patients are like, sure, I'll just do anything, just give it to me. After they've been through or the ringer for a little bit, they don't always feel that way. So again, we're trying to really think about, this is all patient-centric. It's not just about what, Ross wants to do to see what's cool and interesting. That has been a journey for me. It's patient-centric. We really try to say there is a middle ground between walking a new path, which you need to do in cancer. Let's be honest. There's a lot of tumors out there. We need to rethink what we're doing. And thinking about, this is a human being. We're not going to experiment on this person except in a really responsible way. There is a middle ground here, and we really try to hit that middle ground so that, the doctor and the patient can feel like, okay, these people are responsible. They're not just tossing crazy stuff at me. So that's a little bit of a rant, and I apologize for that. But I think for me, it's really important that the patient know that we do take these things into consideration. We're not just trying to throw crazy stuff at them and see what works.
No, I mean, that probably, probably in the lab, you might try a few extra things on the flies, just out of
Oh, that's different.
and that is exactly what that space is for, is
Yeah, exactly.
let's just experiment on the flies.
Mhm.
But when we're coming down to
I
you
am.
team making recommendations to an oncologist for a patient, you're going to be using well-vetted therapies that have some evidence that they can work together well, and that they're safe, and that they're right for this person.
Sure, absolutely.
Yeah, that's wonderful. Yeah, I'm excited to see where this all goes. Again, just in the years since we first chatted, and now, so we'll say 3-ish, 4-ish years, um, you folks have certainly come a long way, and you've really enhanced your process, and the data that you've gathered will only help to make it faster and easier for you to help other people, which is really exciting.
Yeah, no, thank you. I really appreciate it. And it's for me, it's been an incredible journey because Laura, the CEO of the company, she has so I get to play in this sand pit and try new things. And as you say, do things to flies that maybe I would definitely not do with people. Um, and that's lovely. But Laura has been the person that has really taken this technology and made it grow up and really refine it in a way. And of course, this is how that's supposed to be, right? And that is her expertise in doing that. And the team that they have at Vivan and working through this. And like I said, I'm not going to go through the technological improvements they've made, but they have been substantial. And I have learned as much as I've taught for sure on this point. So for me, it's been actually pretty cool.
I bet it's for somebody with your area of expertise and knowledge base, being challenged in this way, that's what it's all about, isn't it? That you're constantly learning and growing and discovering new things. And then in your role at the University of Glasgow, is that where you said you're at?
I am.
Yeah. So you get to, which is so exciting. Congratulations on that, by the way. And you get
You.
to really inform from the front lines, the leading evolution of cancer care here. You get to inform young minds and help them to understand what's possible. And again, that chasm between what is possible and beyond that, versus what is typically done right now. There is such a chasm, and for you to in your work be shortening that gap for individuals who need help now is amazing, and so important. But then also for the young minds who are interested in personalized medicine or cancer research or things like this, to just be hearing from you, from where you started, and again, the combination of yourself in the lab and somebody like Laura with the computer, you know, like that's where
Mhm.
it's at. Absolutely, is that combination of people who understand kind of the molecular elements of the body, or the flies in the lab, and how to manipulate those, and then the folks who can really use that information. Because as you said, and I loved how you said it, if I can find it in my many pages of notes here, the blizzard, what was it, the blizzard of information.
Yeah.
We have so much information. And when we do our tumor testing programs for patients, we test for the 550 for DNA mutations, and then we test every gene for the RNA expression. So that's like 21,000 genes we're looking at. That's a lot of information. Um,
Hmm.
and that all, and of course we have to kind of whittle all that down to the most that, you know, which are working kind of in tandem or against each other, you know, and just as you're doing with the flies, which treatments then are going to either kind of hold the cancer at bay or dial it back, or which, if we target this particular mutation, that might leave these ones to run wild, and we don't want that. And so the combinations that you're getting to explore in real time with the flies, you are actively preventing patients from having to find that out for themselves, essentially. Oh, I should have added this one, or we should have been mixing these ones together. You're saving the patient from having to go through all that experience and letting the flies go through it for them.
Definitely. And so just sort of, and I'm more than happy to have it say a few words about the youth thinking about maybe going into STEM. I've got lots to say about that.
Great.
Um, but first, let me say that as you say, right now is, I know we always say this as scientists, but it is absolutely the case. Right now is an interesting time in the cancer field and in biology in general, because new tools have come online, first of all. So the technology, we've now gone truly to big data. Been a lot of talk about big data. We're there. And of course, having things like AI is going to be key, because I cannot wrap my head around the literally hundreds of thousands of data points that we gather from a single patient. It is wild what comes in. So that's first of all. And as I said, it's not even just random data points. There's spatial information. It's wild. But what's really interesting, to your point, is that, so Laura and I are, I think, a really good example, Laura Tower and I, which is that we work closely together. We're actually quite close in a lot of ways. And that bridges, so I'm, of course, a really basic researcher who works things out into the lab and took a step into the clinics. But Laura is really a bio-entrepreneur who really thinks about, okay, that was great, Ross, but now we actually need to make this more practical. We need to make it more user-friendly, and we just need to make it better. And there's a lot of back and forth. So I work very closely here in Glasgow with clinicians in the hospital to get patient tissue that we can then get information from. And so we're contributing to this blizzard of information, and we're digging deep into it. And then I go and I talk to Laura, and we talk about, well, how would this impact, you know, the technology that we have that's forward facing to the patient? And so that flow of information back and forth, which everyone has always talked about. So in 2024, that is a fact.
That is
That
happening.
happened. And the learning goes in both ways. So just to finish to your point about youth, if you're thinking now of like, what do I want to do with my life? First of all, I can totally recommend getting into science, because first of all, it's like being an artist, but they pay you. You get to play with all sorts of cool toys. If you've always been the kid that's been sort of odd personality, or maybe not super, you know, the most socially adept, come on in, because we got a whole world of people like that, and we love each other, and it's all good. But also because, similar to like the information age in the 80s and 90s, that now we all have iPhones and all sorts of cool gadgets because people were passionate about that. Now that's where we are in biomedicine. We're right at that sort of biomedical age, where now all these cool things are starting to spin forward. I mean, the flies are an example of that, right? And that's all beginning to spin out of, you know, decades of hard work by lots of people. And now we've hit that sort of tipping point where now we've turned to the consumer, which in this case would be the patient, and saying, okay, we've got this whole world of stuff. Now we can begin to aim it to you. And I think Vivan is a great example of exactly that. That's exactly what happened. Is Vivan spun out of all this information coming, and we thought, well, what are we going to do with this? And so our shot at this is, well, here is a classic animal that's been worked on for, you know, 120 years. Much of what we understand about the human body actually comes from this fruit fly. Actually, many or most of the cool names of proteins actually named in flies. And I've been fortunate to be part of that group.
Hey.
And now we're going to bend that to the patient and say, all right, it's been lovely. It's been a great ride. Now, what can you do for the patient? So I think it's exactly as you say, it's an interesting time. And certainly for those who are thinking about what do I want to do with my life, you want to do something cool and important and all of that,
Mhm.
come on in, we've got plenty of room.
Excellent. Thank you. We will make sure that that CTA to the youth gets out there.
Yeah,
Yeah,
yeah.
but that is really the point, isn't it? Is that it's not just a thought experiment anymore or a lab experiment anymore.
Mhm.
The combination of yourself, Ross, and Laura, with her kind of computer savvy, and as you say, the bio entrepreneur bit, and your experience in the lab. They do keep informing each other. It's like, well, why aren't we doing this? Oh yeah, why aren't we doing this? Or could we do this? I guess we could, and that's the, I'm sure,
Well,
exciting.
you've been in our meetings, because
Well,
that's
we have
exactly how they go.
meetings here, right? Where I say to Alex, "Hey, why aren't we doing that?" And he says, "Well, no, but that's not being done." And I'm like, "Well, why is it not being done?" Yeah, why not? And then he's off to the lab and creating the thing, and it's super.
Michelle, you should totally come on in our meetings because this is
I
exactly
I am happy to be a fly on the wall
right.
meeting for sure. Okay, is there anything else? And maybe we leave our talk today with, if you're willing, and we can edit this out if not, but what if there was something that you could say, and it can be a long thing or it can be 3 words, if there was something you would say to a cancer patient right now, based on what you've seen and the knowledge that you have, everything that you understand about cancer care and what's possible, what might you say to a cancer patient at this time?
So I would say to a cancer patient, first of all, I've had the privilege to interact a lot with cancer patients over the past really 8, 9 years when we really began to dig into this. And first of all, I've come to understand it is a scary time
Mhm,
for sure because you don't know what's coming next.
mhm,
And so that's first of all. And it's important that you work with a doctor and you work with others to develop your treatment that are thinking about the specifics of where you're at. So Vivan provides an essentially access to the realization of all the cutting edge stuff that's happening now, as we had discussed. And
mhm,
I hope that was a simple way to put it, which is we're basically bringing all of that knowledge that has been pouring into the cancer world. And we're basically bringing that back to the patient in a simple, accessible way.
mhm,
It will definitely not cure everybody for sure. And I think nobody can make that sort of promise. Every tumor is different. They all run a different course, and we've certainly learned that. We have had some successes. We've published those successes. They're out there for people to see. We have definitely failed with some patients as well, and we're trying to understand those and we're always getting better each time.
mhm,
But this is really sort of in its way, a common sense approach to taking the blizzard of information or the fire hose of information that has been coming at us all and turning it back to each individual patient.
mhm,
For me, this is a very sensible way. And if I had cancer and there wasn't an easy, standard of care should always be tried first. But if that's not working, which it often doesn't, personally, I would go to this because to me, this makes sense. And I think a lot of people think this makes sense. Some people think it's a bit crazy. But I think it is a reasonable way to bring technology back to the patient.
mhm,
That's the purpose of it. I think it's being done very sensibly and with a little bit of pushing the technology forward.
mhm,
So if I had to bring anything to the patient, I would say it's a hard choice. There's a lot of sort of things going out there, and we don't know the perfect way forward. But this is one reasonable, definitely cutting edge way to take technology, push it forward, and push it back to your advantage, and treat you as an individual in your tumor as an individual. So all of these are basically why Vivan was built for this.
Yeah, and again, your process just keeps getting refined. You're able to offer more options faster to patients, which is
Yeah,
see the real
exactly. As the tech, so I am the Regius Professor of Precision Medicine. I do have my finger on the pulse of what's going on in this.
right.
For those Americans like myself who had no idea what that is, it's a royal position. Um, and um, and so, yeah, the truly latest and greatest technology will always rotate back to it. This is actually the point of it. And the
Yeah,
delivery model, which is the fly, is flexible enough that as we learn more, it learns more. It gets better. Absolutely.
that's it. And you have the freedom and flexibility to be constantly evolving your own process as the data comes in. So it is absolutely individual patient driven, this process. And then again, the kind of the culmination of various patients and their data helps to inform your care of the next person that comes along, and more so. And that's, I think, exactly what personalized cancer medicine is meant to be, for
Yeah,
sure.
couldn't have said it better. I agree.
Mhm. I do talk about this stuff quite a lot, because it's definitely become my passion to exactly as yourself to um, be using the most current technologies from all the fields to inform individual patient care and make sure that they and their doctors have the most accurate data to work from, and no time is wasted, really.
Yes, absolutely. And actually, you reminded me, you made a key point, which is we do work with the patient's oncologist. We do work with their doctor. That's also another important point, which is, yeah, you should feel comfortable with the team that's built around you. So
Yeah,
yeah, keep that.
exactly. The whole idea here for us as well is to come alongside the patient and their doctor, in support of their care. We're not taking over, you're not taking over, but you are adding some very important and very personalized, specific to the patient information that, will help them dial in the therapy they need better. And the combinations, as you say, because it's rarely just one drug, and finding the right combination is tricky.
I
Yeah.
love, yeah, that's the brilliance of flies. They'll find it for us.
And again,
So,
you
yeah.
100,000 mice would take a long time to grow up, and where the heck do we house them, and per person, and all the stuff. And again, then you can only manipulate 2 or 3 genes at a time versus 10 to 15. So sounds like you've definitely, you're working with the right critter, that's for sure. And the data seems to support this.
Yeah, and I should say, coming out of New York, I don't want to see another 400,000 mice for sure.
No doubt. Yeah. Well, thank you, Ross. I really can't thank you enough. Again, I'm feeling like from our mentioning of Laura, I probably need to have a chat with her too if she's willing, because she deserves to be recognized. Uh,
Yeah, it's
Absolutely.
amazing.
Laura Towert, call out to you for everything that you're doing for cancer patients, for biotech, for all, and for Ross. Kudos to you, really. I know it's not easy for sure. But hearing from the kind of the computer side of things, I think would really benefit people to understand kind of what's happening developmentally there. So I'm just dropping a little pitch in your general direction to maybe see if Laura might like to have a chat with me too.
I'll let her know. I'll let her know. I am a genuine big fan of hers, which is why we connected for this whole technology. She's impressive.
That's my sense too of the brief conversations, but she's in Uber on her way to this and there when I get a chance to speak to her.
And that.
So busy. But yeah, really understands again the scope, and people who can kind of see the big picture, they're precious and rare. So we
I
celebrate them.
agree. I completely agree.
Yeah. Well, thank you so much, Ross. I really appreciate.
Michelle, this was a ton of fun.
Well, how interesting was that? What incredible technology, and what an interesting mind and human being is Ross Cagan in terms of his experience working with genetics and cancer patients. But this, unique modality that they're using of these flies and the fly armies is such an interesting concept. And of course, as you heard, they're just continuing to refine to make it easier and faster to figure out which molecular features to genetically program into the flies so that they can get results faster for patients, but also they can start to work with all different types of cancers as well. So very exciting stuff. I will update you as things unfold for the Vivan team for sure. I really hope you enjoyed this interview. And please do, if you have any questions for Ross or for myself, please do just make a comment in the chat below. I will be happy to follow up with you and get your questions answered. That's what this is all about, making sure that you are as well educated as you possibly can be about your cancer and about how best to treat it, but also about some of the new exciting technologies that are coming. There's always something new. That's what I most want you to take away from this. This is just one of many modalities that is brand new and that cancer patients can use to enhance the quality of their care, to enhance the number of treatment options they have. And me and my team at Cancer Treatment Options and Management and at the Cancer Just the Facts program are here to help you figure out what's best for you and how to get access to it. So let us know how we can help. And again, post any comments or questions in the chat specific to Ross and his team, and I'll do my best to get them answered for you.